Hello everyone, I am Mukaram Bhat and I'm very excited today to share with you my favorite topic in science, that is mitochondrial trafficking in neurons. Before I would start, I will give you a brief introduction about my topic. Then we will see what is meant by this mitochondrial trafficking in neurons. Then we'll see what are the proteins that are involved in this mitochondrial trafficking. Then we'll see if there is the impairment in this mitochondrial trafficking, or if somehow there is a blockade in this trafficking, or if there is an abnormal trafficking, what it leads to, or what are the consequences of that. So first we would talk about neurons. So neurons, they are like pretty complex cells, and they are the cells that are important for our brain to function. Now as I said, they are highly polarized cells, right? So, or pretty complex cells. So they consist of these complex processes. These are dendrites, relatively small cell body, or soma, a pretty long axon. So this axon can go up to 2 meters in humans, or even more than that, and this ends up with the synapse. Now what is the synapse? So this is actually the talk between the two neurons, right? So now when the two neurons are able to talk with one another, when the first neuron talks with the next neuron, so there is something called signaling from one neuron to the next neuron. When that signaling is happening, then only your brain is able to function. So if that signaling will stop, our brain will not function. So as I said, they are really the key for sending these signals across our brain to help us with the everyday functions like talking, walking, right? So now for these neurons, right, to maintain their function, they need energy, and this energy is provided by the mitochondria. And we know that mitochondria is a double membraned organelle, and what it consists of outer membrane, inner membrane, the matrix, and in between the outer membrane and the inner membrane there is the intermembrane space. And we know there is electron transport chain which is present on the inner mitochondrial membrane. What it does, it produces the energy in the form of ATP, and this ATP is utilized by these neurons. Now we will see why mitochondrial trafficking, right? So why we need this mitochondrial trafficking in neurons. So as we know, mitochondria is present in all cell types, but neurons, so they are highly enriched with mitochondria. So if we talk about our brain again, right, so it has many cell types, but these specialized cell types, so that we call neurons, so they are important for our brain to function. And we know the mass of the mammalian brain, it accounts for 2% of the total body weight, and energy demand of the same brain is 20%. So 20% is a pretty high amount of energy, right? So 20% of the total energy. So it has been said that brain is called as selfish organ of the body, and it's correct to call it the selfish organ of the body, because brain is not going to compromise on the energy, right? So other organs, so they may compromise on energy, but brain is the organ that's not going to compromise when it comes to energy, So now this energy is utilized by the specialized cells, so that are called neurons. Why? To maintain the synaptic activity. What's this synaptic activity? It is the talk between the two neurons, right? Or multiple neurons, not only two, but two I'm saying here so that you can understand, but these can be multiple neurons at a time, and that is maintained by oxidative phosphorylation, right? So that is the process happening in mitochondria, and by that what we get ATP. Now if we talk about this mitochondrial trafficking, why we need mitochondrial trafficking in these neurons? So as I already said, that neurons are like pretty complex cells, and we know the biogenesis of the mitochondria, it takes place in the soma or the cell body of the neuron. And then what we need to do to maintain the energy homeostasis in these regions, so we need to traffic that mitochondria from this place up to this place or this place, right? So this is presynaptic photon and this is axonal terminal. Now what we do, so we need to equally distribute that mitochondria from this place to this place, and that is happening by the process that is called as mitochondrial trafficking in neurons. And as I already said, so that trafficking is required to properly distribute mitochondria in these axons, so to maintain the high energy demands, right? So these are the high energy demanding regions, so be it presynaptic photon or axonal terminal. Now we will see how this trafficking is regulated in neurons and what are the proteins that are involved in this phenomenon, right? So the players that are involved in this mitochondrial trafficking, or the proteins that are involved in this, so actually there are many proteins that are involved in this mitochondrial trafficking, right? And that itself is regulated by these proteins. So first there are motor proteins, right? So it can be kinesin and dynein. Now kinesin plays the role in the anterograde transport when we have to traffic the mitochondria from the cell body up to the axon, right? So then there is a motor protein that is kinesin, so it moves the mitochondria from this place to this place. So this is called a plus end directed motor. Why? Because it is moving towards the plus end. Now dynein, so we have now mitochondria here, right? So we have trafficked the mitochondria previously, right? Now this mitochondria is depolarized or irreversibly damaged, so it needs to be like trafficked back to the cell body. Why? Because the degradation machinery is in the cell body, so we need to degrade mitochondria, so it has to traffic back to the cell body. That's why dynein motor here. So it is the mindset directed motor, right? Dynein. Now there are also adapter proteins involved in this. Now what are these adapter proteins? So what they do actually, they connect the motor protein with the mitochondria itself, right? So these adapter proteins can be like track 1 and track 2. So this has been seen in the mice model, track 1 and track 2. And there is also Milton that has been seen in the Drosophila. And there are also anchoring proteins. So what anchoring proteins does, they anchor the mitochondria into the adapter protein, and that adapter, as we can see, that's itself binded to the motor protein. So we can see this whole as a complex, right? Motor protein, anchoring proteins, and adapter proteins. So if somehow there is like loss of any of these proteins, so we will not properly, will not be able to properly traffic mitochondria either in the anterograde direction or in the retrograde direction. So there is a bidirectional transport happening here. So if there we have the loss of any of these protein, so what will happen, there will be the disruption in the trafficking, and that leads to pathology. So that we will come to know in the next slides. Now, as already said, the mechanism, so how this mitochondrial trafficking takes place. So there must be some mechanism, right? So where we have to traffic the mitochondria from the cell body where the biogenesis takes place to the high energy demanding region. So high energy demanding region can be the axonal proton or synaptic terminal, as I already said. So how this trafficking takes place, we will see, right? So first we know, as I previously said, that kinesin or KF5 motor protein, it is moving on the microtubule. Now microtubule is like a track, right? So I will give you an analogy here that microtubule is like a track. This motor protein is like a train, right? And this all is the passenger in the train, right? So that whole is now the complex, right? So if we don't have the train, suppose let's say we don't have the train, but we have the track and we have the passenger, so will it be like will that passenger be like able to get the benefit of that that thing out there? No, because the train, we are missing here train, so there is not like any significance of that, right? So same, if we lost any of these proteins, so there we can't like then further traffic the mitochondria, right? So as I already said, so this is the placenta directed motor. What it means, so it traffics the mitochondria from the cell body to the synaptic terminal in the anterograde manner, in the forward direction, I can say. And attachment of this kinesin to mitochondria, it's mediated by anchor protein, that is myro in this case, this, and it's adapter protein that can be like track 1 or track 2. Now, how this trafficking is regulated. Now we are at the spot, so we arrived at the destiny, right? So now we need to like stop the mitochondria. So how this happens, so there must be some mechanism. So they are like stopped in the calcium dependent manner. So what happens is, so on the myroprotein there are two calcium binding EF hands. So we know that when the signal passes in the neurons, right, from like, so let's say from top to bottom, so there is a reversal of charge on the plasma membrane of the neurons or the cell membrane of the neurons. So when there is a reversal of charge, so we know there are calcium ion channels on the plasma membrane. So when they sense that charge, so when the charge gets flipped, they sense that, and they are now open since they are voltage gated calcium channels. So when they are open, there is a rush of the calcium into the neuron. So when there is a rush of calcium into the neuron, so what happens is, here we have the myroprotein that has the calcium binding hands, right? So when when the calcium binds to this myroprotein, what happens is there is a conformational change in the whole complex. So when there is a conformational change, the motor domain that was previously like bound to the track, right, the microtubular track, so it gets disassociated from the track and it binds to the myroprotein, right? So motor domain binds to the motor myroprotein, and when this happens, the mitochondria is stopped at the like destiny where there is a need of the energy. Now, so this was about the kinesin or KIF5 motor protein that was moving the mitochondria in the forward direction. Now we know that we have mitochondria here, right? So in the axonal terminal or axonal bouton. So what happens is that we know that everything has a turnover, right? So in nature everything has a turnover, so does mitochondria have, right? So we know that mitochondria after some time it will like become dysfunctional, or it will be like irreversibly damaged, right? Or its membrane potential is it loses its membrane potential. Now what happens is, so it needs to get back to the cell body in order to get degraded. So because it's in the cell body where degradation machinery is, and there we have the retrograde transport that is like mediated by the dynein protein. So as we can see in purple color, these are the dynein motor proteins. They move mitochondria back to the cell body for degradation. Now as I said, so this was the transport back to the cell body, and before that we learned about the transport that's happening to the axonal terminal. So if there is any disruption in trafficking, right, be it like in the forward direction or the backward direction, so what happens is, so it leads to pathologies, right? So when there's an impairment in the trafficking, or when there's a blockade in either of the trafficking, either like anterograde or retrograde, so it leads to motor defects and neurological defects or pathologies like neurodegeneration in humans. So it has been seen that in Alzheimer's, ALS, or Parkinson's disease, so if there is a disruption, so it leads to these diseases, right? So the take-home message is that axonal terminals, so they are highly energy demanding segments of neurons. So when these energy demanding segments, so that is fulfilled by the mitochondria, and mitochondria, so we need to traffic the mitochondria to the terminal part of the axon, because that's the high energy demanding region, and that happens by mitochondrial trafficking. And there are different proteins that are involved in this mitochondrial trafficking, be it myro, track1, track2, kinesin, and dynein. So mutation or loss of any of these proteins, what it does, it leads to the mitochondrial trafficking defect, and defect in the mitochondrial trafficking leads to motor defect and neurodegeneration. So it has been seen. So thank you for your attention, and with this I will conclude my presentation.